This video will explain how and why some mushrooms are not metabolized by the human body and when ingested can lead to severe complications such as organ failure and death. The main character of this illustrative video is Amanita phalloides, also known as the death cup. This mushroom causes hundreds of deaths worldwide. It is one of the most famous and lethal Amanita toxin-containing mushrooms. These amatoxins are responsible for over 95% of deaths caused by mushroom poisoning worldwide. But why is that? When this mushroom is ingested, the toxins survive the digestive tract and cause cell necrosis in the gut and in the liver, as the deadly mushroom contains two specific toxins, the phallotoxins and the amatoxins. Phallotoxins are responsible for damaging the endothelial cells of the intestines, also known as the enterocytes. However, they are likely not absorbed by the intestine and therefore cannot reach the liver. Then, we also have the amatoxins. The amatoxins, specifically alpha-amanitin along with beta-amanitin, are the ones that mainly harm hepatocytes. These toxins are not degraded at high temperatures, for example when a mushroom is cooked, and are conserved at low temperatures, so freezing also does not eliminate the toxins. Furthermore, these toxins are also enzyme and acid degradation resistant, thus they don't undergo any metabolism processes such as detoxification. After surviving the digestive tract, these amatoxins enter liver cells where they inhibit RNA polymerase II. They inhibit RNA polymerase II by binding to the bridge helix, which facilitates the translocation of DNA for each nucleotide addition cycle. It also binds to the part of the complex that is adjacent to the bridge helix while it is in a specific conformation, which locks the RNA polymerase into this conformation. This dramatically slows down the translocation of DNA in the RNA polymerase. Inhibition of RNA polymerase II leads to the inhibition of mRNA synthesis, which is essential for protein synthesis. Without protein synthesis, cell metabolism stops and the cells undergo lysis. So, here is a short recap of what happens in the hepatocytes. Firstly, since RNA polymerase II is inhibited, the cells no longer synthesize mRNA, meaning the ribosomes are also no longer translating mRNA molecules into amino acid chains. This consequently leads to the inhibition of protein synthesis. The cells no longer produce essential proteins, causing the cells to undergo lysis. However, that's not all. Due to cell stress and inhibition of protein synthesis, the p53 protein, which detects changes in RNA transcription, is activated. Then, it binds to the anti-apoptotic complex in the mitochondrial membrane and enhances the release of cytochrome C. Cytochrome C induces the cell to undergo apoptosis. The accumulation of alpha-amanitin also leads to inhibition of the antioxidant enzyme catalase, which is responsible for decomposing ROS. Accumulation of ROS can lead to oxidative stress, which can in turn disturb DNA metabolism by damaging nucleic acids. Symptoms of Amanita poisoning only occur when significant damage is already done. Therefore, clinical signs are only noticeable several hours to several days after ingestion. These symptoms usually start with severe vomiting, diarrhea, and abdominal cramps. Other symptoms may occur as well, such as headaches, dizziness, and coughing. If the patient is somewhat hydrated, they will enter the so-called honeymoon phase, a phase during which the symptoms subside. The patient feels better for a day or so, but the poisonous toxins are still hard at work inside the body. Usually, the diagnosis is first based on the symptoms. Often when patients with Amanita poisoning symptoms show up to the ER, they or their acquaintances will inform the doctor that they have ingested some type of mushroom. Based on this information, along with the symptoms, the doctor will order several tests to be done, such as a chromatographic assay. Treatment of Amanita poisoning starts with providing standard support. This includes a stomach wash and laxatives in order to remove any remaining toxic product from the gastrointestinal tract. Then, penicillin and silibilin are administered to inhibit cellular uptake of the toxin. Additionally, activated charcoal hemoperfusion is administered to remove the toxin and to replace the coagulation deficits. As a last resort, liver transplantation is performed. With fast reporting and proper medical care, 85 to 90% of Amanita poisoning patients survive. That concludes our video about the poisonous mushroom Amanita phalloides. Thank you for watching.